Okay, welcome to the webinar at Ona. My name is Joao Vendramini, and I am the forage specialist here at the Range Cattle Research and Education Center. Today, I'll give you a brief update on the Limpograss project that we have been conducting the last 3 years. And I will start with a little introduction about the Limpograss because that is not a cultivar or a forage species that is widely used. It's the use of Limpograss is confined to mainly South Florida and some other parts of the world that are wet and tropical. And Limpograss, it became very fast the second most used forage species here in South Florida. Primarily because it can hold some, it can survive in places where they have poorly drained soils. And that's why it does well here. We have a lot of those poorly drained soils. And also because it has a little different characteristic than other warm season grasses, so we can keep the nutritive value and the digestibility even when it's a late maturity. So most of the warm season grass, when it gets too mature, the nutritive value just decreases sharply, when the Limpograss will maintain the digestibility over time. So we really like Limpograss to be used as our winter forage for stockpiling. In fact, this picture that you see on the screen, this is a picture taken in January 2007. So you can see how Limpograss can do during the winter there. Just a brief history going back to the Limpograss species. This yellow line is the border of Mozambique here in South Africa. It's where the Limpograss comes from. It's the southern part of Africa. And this is a picture of the Limpopo River. So this grass does really well close to the banks of the river here. And that's where it came from. In 1964, they brought some collection from there to the United States. They used to bring all the collections and send up north to a central collection place. And then Dr. Quisenberry, in around 1980s, he was there and he collected some of these entries that were brought from Africa to Florida to test it. And then he released about 5 different cultivars, when most of them disappear over time for different reasons. But Floralta was the one that was the most persistent under grazing. And at the end of the day, Floralta became the main cultivar that we use here in Florida. In 2014, after 6-7 years of selection and breeding, so we released 2 new cultivars. It's called Ken High and Gibituck. Those are hybrids between Floralta and Big Alta. Big Alta is one of the 5 cultivars that were released in 1980, but because it didn't persist under grazing, Floralta just disappeared over time and Floralta took over. So the cross between Floralta and Big Alta made those new cultivars there that is called Gibituck and Ken High that were released in 2014. So as I said, 5 years of selection here at Ona and in Gainesville. We collect data, not only clipping data, but also grazing data here. And those forages, before they were released, they went at least 3 years under clipping and 3 years under grazing at different intensities. The main goal of the crossing was to produce forage with better nutritive value. That was the goal. However, I think what we end up with the Gibituck and the Ken High, they have forage that produce more than Floralta. And on the top, we have seen that this Gibituck has been more persistent under grazing than Floralta. So and I will show you some data related to persistence. That's why I think we like it, because one of the biggest problems of Limpograss over time, it was persistence. People plant Limpograss pasture, sometimes doesn't manage very well, and that became a Bahia grass pasture over time. So then this Gibituck, I think, is a very good fit for what we do in beef cattle with a low management or very low input system. And in addition to that, in this project, we have the fertilization component. And here in Florida, fertilization has been anywhere in forage production systems, fertilization is the most costly input. We know that. Our money goes to fertilizer, although I think it's a money well spent if you think about production and persistence of your grass, but it is costly. And here in Florida, it seems that we have a bigger challenge than in other places, primarily because weather and soil. These sandy soils doesn't hold nutrients, so what happens is we lose a lot of our fertilizer because of heavy rain and sandy soil. Sometimes it's too heavy, the rain, and then we lose everything. That's why we haven't seen the responses to fertilize that we see in the literature. Sometimes here, the responses that we get, even for nitrogen fertilization, is quite variable because of this weather condition. So as I mentioned, we started this project in 2016. It's a Florida, funded by the Florida Cattle Enhancement Board. It's a 3-year project. And it was because those new cultivars were tested under a little higher input system. When we released them, we used to fertilize them every 6 weeks. So then I would like to test how those new cultivars will do under low input systems or low fertilizer levels under clipping, and also to have a little better idea of how those cultivars behave under different clipping regimes or regrowth intervals. So that project was, as I said, started in 2016, and we did in 3 locations. The other thing is we have a lot of interest of Limpograss in other parts of the state, although there is a perception that Limpograss doesn't take cold weather. So if you go north of I-4, we will not see a lot of Limpograss. But we have this project in Gainesville and Mariana, and you have seen that that's probably not the case. I think management and where you plant the Limpograss is probably the most important factor in order to make those plants to be persistent, even in Mariana and Gainesville. So in all 4 places, we have the 4 cultivars, Floralta, Gibituck, Kenhai, Entry 1. Entry 1 is one of the hybrids that we didn't release. So it was promising. The production was always intermediate. It was very persistent, but never made the cut for release. So we decided to investigate further Entry 1. And we have 2 fertilization levels. There will be the low and the high. And then we have 2 harvest frequencies, 6 or 12 weeks. So in previous work done here by Dr. Silveira with fertilization on Limpograss, on Floralta Limpograss, we noticed that harvest regime or frequency was even more important than fertilization towards the persistence of Limpograss. So that's why we added that factor to test not only the fertilization, but also how that will affect the grass when it's related to the frequency. Although, as I mentioned, the nitrogen efficiency data that I will show you is actually the nitrogen that we applied in relation to the herbage accumulation that we had, okay? It's not the total nitrogen that the plant uptake, it's the nitrogen that we apply. So the calculation is nitrogen applied divided by the total mass. And as I mentioned, we have the data collected in all 3 locations, but for the sake of time today, I will just present the data that was collected here at Ona in 2017. So now I will switch to the results, and I have a lot of numbers here. I will go slow, and I would like to ask you please to focus on the square. And then for the herbage accumulation here, or the herbage yield, if you want to call, and this is the yield for a 12-week interval, okay? It's 1 12-week harvest or 2 6-week harvest, okay? We have seen, as some of our previous research show, how Gibituck has produced more than the other entries, when there was no difference among Kenhai, Floralta, and even Entry 1. It was pretty much the same. With the greater production, that pretty much translates in better nitrogen use efficiency, because that plant produced more with the same amount of nitrogen. So even in the lower levels or higher levels, it's more productive. There was no interaction. So that plant, if you fertilize less or more, will produce more herbage. However, we didn't see any difference in nutritive value among cultivars. We have noticed some differences in digestibility in the past, but it's not consistent. So we have a forage here that will produce more, but actually the nutritive value will be quite similar. Going to the regrowth interval effect, and that's what I mentioned at the start of my presentation, how this Limpograss is different. Look at the difference between 2 6-week harvest and 12-week harvest. How much more forage we can have here if you let that forage to grow to 12 weeks. So longer regrowth interval in this Limpograss is not a bad thing. Sure, we pay the price here. There is a decrease in nutritive value, but see the magnitude that we see in the decrease in nutritive value is actually small compared to the amount of forage that we grow in that interval. So this grass behaves very differently. And this longer regrowth interval should be considered, even if you have to pay a little the price to have a lower nutritive value. And then it results in better nitrogen use efficiency. Actually, it's a better investment for your nitrogen fertilization because you're going to produce more forage if you let this grass to grow in longer regrowth intervals. And finally, here is the fertilization levels that I mentioned, the 2 levels, high and low. There is a little confounding here because we have not only nitrogen, right? We have nitrogen, phosphorus, and potassium, N, P, and K in the formula. But that was designed actually just to have the same amount proportionally in both treatments. But you see here, we doubled the fertilization level, but we just increased 1,000 pounds to the acre of dry matter here. Look how the increase is really small. And again, this is 3 12-week harvests during the year. Think about it. This is the average of 3 12-week cycle, okay? So then it's quite consistent that we produce a little more with greater fertilization, but look at the nitrogen use efficiency. We fertilize much more, but we decrease the efficiency of the fertilizer that we put in. So then we'll go further on our calculations in the next slide or so, and we'll talk a little bit more about this efficiency here. But the other thing that is quite interesting in the literature, you see you put more nitrogen in these warm season forages, the nutritive value increase, at least the protein increase. Look at what happened to the Limpograss. No difference in nutritive value. We didn't even increase the crude protein concentration, adding more nitrogen. So it's a different grass when it behaves towards maturity and fertilization. So in this table here, what I have is the signs of persistence after 2 years of harvest, okay? This was evaluated at the end of the trial in the end of 2007. Look at the root mass of Gibita compared to the other forests. This entry 1 didn't differ from Gibita statistically, but it didn't produce much, right? So that may be the case. That grass doesn't grow much upright and the tops, and it produces more growth underneath that we don't see and save itself. But the Gibituck, it grows better on the top and the roots. And then you see here in the ground cover, it is the most persistent among all the cultivars. We still have 80% of the ground cover compared to the other forages here. More root mass, more ground cover. So it's more persistent, it produces more, and it is more persistent than the other cultivars that we have. Okay. And then the regrowth interval also had some effect on the root mass. You let it grow for 12 weeks, look at the root mass. Increase because the plant has time to recover. And then look at the ground cover on the 12 weeks. So 6 weeks, that's supposed to be a long regrowth interval for stargrass, Bermuda grass, Bahia grass. It seems not enough for Limpograss. You do 6 weeks, you have a harvest, a forage with a little better nutritive value, but it doesn't persist. That may not be recommended for this Limpograss to harvest so frequent. And then finally, You see here the effect of fertilization. You put more fertilizer, you have more roots. And that is one advantage of increasing fertilization levels that we don't see. We just want to see how many bales of hay we harvest, right? Or how many cows we put per acre. But at the end of the day, greater fertilizer results in more roots and better cover. So that is probably an effect of fertilizer that you just see long time. Over time, you actually find out that you didn't fertilize enough when you have a stand that is probably 50% Limpograss and Bahia grass is taking over. So fertilization is also very important, not only for roots, but also for the ground cover and persistence of the grass. And then I made some calculations regarding these fertilization levels that we apply. As I mentioned, we produce more, right? When we put a little more, when you put more fertilizer. And that is the nitrogen concentration that we have in the grass. So this is not a grazing system, this is a harvest system. That means everything that we cut it, we remove it from the plots. This is the amount of nitrogen that we remove after every 12 weeks, okay? So here you apply 80, you remove 55, here you apply 40 and you remove 40, right? So what we saw in the previous slide, that when we are removing everything that we are applying, what's gonna happen? You're probably gonna lose the stand. When you apply the 80, you have a better cover. You have some nitrogen residual here because you are not removing everything that you are applying. So that is the bottom line. So, and we're gonna discuss a little later about it. And the data is consistent across locations. In fact, in Gainesville, we produce more forage with the 80 than the 40, as we did here. But the export of nitrogen was close to the 80, but we still saw some better persistence than when we do 40. And finally, I will show you some of the data that probably is not as practical as that I showed before. So we did some gene expression in those plants, and we did by individual, although we calculate like this table is by treatment or by cultivar. And we have seen that DOF1 is a gene that has been identified as a good indicator of nitrogen use efficiency in corn. And the rubisco is actually the rubisco gene from the Limpograss that was identified in Limpograss. So we just saw the gene expression of this two. We tried 7, but those were not correlated, and we didn't have a good expression even with the primaries that we had. So we selected off in the rubisco because those were the best ones. And then we saw that when you have a lower expression of the gene, we have a better herbicide accumulation or better nitrogen use efficiency. So here the Gibituck had lower and can high than these two. And for the rubisco, the same thing. Gibituck have the lowest expression of the rubisco gene. And when we did the correlation among all plants here for the season between the gene and nitrogen use efficiency, you can see an inverse correlation. So we increase the gene expression, we will decrease the nitrogen use efficiency. And that is the same for the other gene. So we still need to investigate. I think we need to go a little further because there is very little work with Limpograss gene expression. So we have almost nothing other than just identification of some genes like the rubisco that we found. But I think that is preliminary and probably will be a good path to go to try to see if you can select the plants by this gene expression and have that as a tool to help us to select plants. So as the implications of the work and a summary here, I think, have enough evidence that if you have to plant a new Limpograss pasture in South Florida, I would greatly recommend the Gibituck because it has been consistently superior than the other cultivars that we have over and over, probably after 6 years of evaluation. That differ between location. So you may have differences in Gainesville and Mariana. So we're probably going to present that data sometime later. But here in South Florida, it seems the give turkey has been the best, not only because it's the production better, but because the persistence. I would rather to have some grass that will not produce as much, but will be perennial. And that's why we use Bahia grass all our life, right? It doesn't produce much, but doesn't die. So we want to improve the grass like this that also doesn't die. And give turkey is a pretty good candidate for that. We have these greater regrowth intervals, always better for everything. For our production, it didn't help with the nutritive value, but as I mentioned, the magnitude was small. So if you have a Limpograss pasture, I really recommend, and if you harvest, right, for hay or haylage, I think we should consider these longer regrowth intervals because we promote persistence and production, right? And then you can correct the nutritive value with some supplement. So we'll give something for the animal science people to do, is to adjust that supplement and make this grass worthwhile in the winter. It is perceived that this lower fertilization level may give you a better efficiency for the amount of mass that you produce by the amount of fertilizer. And I think there will be a scenario when we have this low cattle market that you are trying to save. You can probably get by with these lower levels of fertilizer when you don't have a budget for one time or twice, or when you have a grazing system. Because in a grazing system, what happens is you have a lot of these nutrients recycling into the system. So if you apply 40 pounds of nitrogen, you're going to have a great portion of the nitrogen coming back to urine and feces. So for grazing systems and for tough times, probably the lower levels will work. But if you are harvesting hay or haylage and you keep doing lower fertilization levels, what's going to happen is you're going to lose your stand. That's why you need to consider to adjust those levels, not only nitrogen, phosphorus, and potassium, and to keep this grass alive and persistent. I think that's probably the bottom line of the fertilization. And the gene expression is a new adventure here, and I think we have some preliminary data that are consistent, statistically different from between species. So that may justify some of this difference in production and behavior that we saw among the cultivars that we have, but still need a little more work to see how consistent will be over time under different situations. So we do have the samples at the end of the trial. Those were samples that were collected when those grasses were planted in the first year in 3 locations, and they were consistent. So now we're going to collect the sample we did and we send to analysis. We still don't have the results at the end of the trial. And we also want to see if management will change the expression of those genes. So for example, plants that were fertilized with higher levels of nitrogen, how those genes will express after 2 years when you keep fertilizing these plants better. So it's all questions that I think we're going to follow up. And it's rewarding to work with Limpograss because probably I know that we will have 2 or 3 people in the earth working with Limpograss, and I may be the only, or me or somebody here in Florida, you know, we're going to be the only experts in Limpograss because it's not widespread in the earth. So, and I would like to remind you about the podcast that we have monthly. So we'll connect here at the website. And we have monthly podcasts regarding issues on cattle production, forage production here. And my contact information is on the top. If you have any questions about the information that I provide or about any topic related to forage, please feel free to contact me. And thank you for your attention, and we will open for questions. The question from the audience was, we sell some plant material here at the research center, and if we eventually visit with the producers after they establish the grass, and what is the feedback? And we do. We do that actually a lot. because not only they wanted to give you feedback, but also see if everything is fine, if you have any suggestions. And for these hybrids, is amazing how is positive the feedback that we have with people that are receiving this Can High and Gibitak from here. Initially, we gave the plant material to 7 producers. It was the first release, but now it's open, and those producers are also selling plant material on their region. So we have it strategically spread around, and I have every time that I go, I have some pictures and I send to Andrea. So, and it's, I think everybody that is trying this new Limpograss, so far we have people that are satisfied with the cultivar, and so far we don't have any bad surprise, because we know that after a while, you know, we start getting some issues here and there, maybe local or maybe based on the cultivar, but so far it's doing very well.
On the dry matter production versus mass potatoes, have you ever looked at ratio of leaf to stem? Are they the same or different?
Yeah, the data is similar to the nutritive value data among cultivars, right? That is your question? Yes, it is a question related to the leaf to stem ratio in the different cultivars. We found in a previous work that the Can High had a little greater leaf to stem ratio, but it's not consistent across locations because we didn't find the same thing here. So, and that may make sense because the Can High had a little better nutritive value at the start, and that may be the reason. However, the Limpograss, as I mentioned, is a very different plant because the digestibility of the stem is similar to the digestibility of the leaf when you go in the first 6 weeks. We found the same digestibility when we did the trial about the leaf to stem proportion, and we did the digestibility separate, and they behaved the same. So, but that may be one reason why they have a little better nutritive value in some instances, better leaf, but that's not consistent. I have a question here. We can go back to the audience here. Any potential for looking to rhizosphere or soil microbial process that could influence or explain the nitrogen use efficiency of the successful Limpograss hybrid? Yes, that will be something important to understand a little better those roots. It's not on my expertise, but I have a student that is coming to do some work on roots. He's a new PhD student, not Limpograss precisely. He'll do on legumes, on Echinopsis, but there are some evidence that this warm season grass, sometimes they can have association with a bacteria and also fix some nitrogen. That's minimum, but it may happen. So that may be another step to see if that better efficiency may be that this plant can actually do something with the association with microbes in the soil. But we have not had any data related to that so far. Another additional question for the gentleman with the blue shirt here.
I'm interested, I know you didn't show it, but since we have some time, I'm interested in what performed best in Gainesville and Mariana.
The questions were about what performed better in Gainesville and Mariana. We still do not have the persistence data in Mariana, but in Mariana so far, we didn't see a difference between Can High, Gibitak, and Floralta. It was the entry one was the lowest production in Mariana. In Gainesville, Can High and Gibitak was better than Floralta and entry one. I think we have enough evidence from the entry one that it does not have merit right now for release. So I think it's something that now we probably know better that probably wouldn't have any merit because it doesn't bring anything better. But in Mariana, it seems that in Gainesville, Ken High always do a little better, little colder weather. And in Mariana now, we didn't see difference, but we just have 1 year of data because Mariana, we start 1 year later. So we have 17, 18 data. So by the time that we finish the 18 data, we will have probably a better idea what was the production in Mariana. Mm-hmm. Yes, the question is about how that gene expression data will behave with things that we can control, like climatic conditions and soil, and we think there will be. It probably does, but for us, because this is a field study, it will be very difficult to detect because all the plants are in the same environment, right? They are all facing the same thing, but I'm pretty sure if you grow a limpograss in a soil, there will be higher fertility, or they'll have a different water regime, right? That will not be flooded 3-4 months of the year. I think that may change. I think that may change actually the gene expression and the behavior of the plant. We do. But we didn't compare locations in any of the data. We just tested the cultivar within location. So numerically, they are pretty similar. The amount of the fold amount for each location. But in Mariana, we saw exactly the same thing. In Gainesville, it was a little different, but we have to consider that the plots in Gainesville were established earlier. So they have been established like for 4 or 5 years, when in Mariana and Ona, they were just established at the year of the research. So that's why I think that, and that's why it led me think the management can change something, because plots that were established, we didn't see any consistent, but for the new plots we did, here in Mariana, they have different conditions. Yes. Uh-huh. 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 We have so little scientific information about limpograss that may be the most cited research that we have. That may be probably the most popular because we have very little information about it. But the thing is, every information that we get about limpograss, it came from the United States. Because in Africa, they have never explored the grass until we brought it here. And then in South America later, they have few places that they have tried. But again, the literature doesn't show them. Some people do something here and there, and they never publish. So that may be the only African reference that we have about the Limpopo River, maybe the Yeah, and that'll be nice because I can put that reference with the picture from the Limpopo River. At least you'll be a picture with a reference. And we have producers here in Florida that they like to hunt. And it's amazing, producers from South Florida, they go to Africa and they want to go to the Limpopo River. So we have people in Okeechobee that went to hunt and they have to go around just to go to the Limpopo River to see what it looks like because of the limpograss. So that's how interested they are and how much they like the grass. Oh, I have another question here before I finish. Is there any difference pertaining to soil pH values relative to the Limpograss as opposed to other Florida grass such as Bahia, etc.? The IFAS recommendation have Limpograss between 5.5 and 6. It's similar to what we have for Bermuda and Star, while the Bahia grass is a little more acidic. It can be 5.5. But we don't have, I think, enough information regarding soil pH and Limpograss production, to make a different recommendation. I think we saw that a little higher pH at 6, the plant performed better, but that may be really well related to the amount of fertilizer that we use on those trials, right? We have a better pH, the grass has a better fertilization efficiency because it has a higher pH. So it may be that if you change the fertilization levels, you may not see a lot of difference. But in general, it's grass that do very well here for us, even in pH 5. But we recommended pH 6 because Limpograss in general, you spend a little more time, more money on fertilizer, and that will increase the efficiency of the use of the fertilizer. But I'm not saying that if you have pH 5, your Limpograss will die, because we will not. But it's better if you increase pH, and the IFAS recommendation is around 6. Okay, thank you.